jump back. We're now kind of in the middle part of the notes. We would have done this in the middle of the unit if we'd been here, but since I knew we were coming back, we moved this to the end because this is where most of the labs for this unit are. Today, we are just going to start talking a little bit about biotech. I added one slide in here today that adds some definitions of kind of some of the major procedures and things that we're going to talk about during class, just so you kind of have an overview of what we're doing this week. And then we're going to start looking specifically at gel electrophoresis and kind of how that works, and that's what you're going to be practicing in a little bit. So the first thing I'd like you to do is just turn and talk to the person next to you, or you guys can kind of talk front and back, and Hannah, you can maybe talk in group 3, either in front or behind. And people online in the chat, please discuss, what do you think of when you hear biotechnology or genetic engineering? What do you know about it? What do you remember maybe doing in 10th grade? Anything that you've heard of, anything you think of, anything you know. Turn and talk about that for a couple minutes. And people at home, please chat.
It'd be like an entire week's of news, and all like piled into a video. Just like that's so weird. Maybe they can like modify DNA.
Okay.
Oh, that's a good thing. Oh, I know that, the sheep.
Yeah. Let's hear a couple of the things that you came up with. Online, Abby said she thought of some line tests that we've done before. I think I know what she's referring to. I think it's one of the things we're going to get back to here. Macy said she thinks about medical engineering, like medical technology. Biotech is big in some medical fields, and it's a big industry in Minnesota. What other things did you come up with? What else were you talking about? We'll just go around real quick. What were you guys talking about?
Like where you can change genes and certain things and like modify the DNA.
Okay.
We talked about how they're trying to use like viruses or like create viruses or enzymes that can go into the body and fix like diseases that have been yet to be treated.
Yep, that's like gene therapy is what they call it. But yeah, cool.
So we can replicate DNA.
Yeah.
So, for example, make a new apple.
Mm-hmm.
Yeah. Take stuff.
Yes. So there are different ways to do that. You can actually go in and change the DNA on purpose. You can also just like breed different types together, which is kind of like old-fashioned genetic engineering because they're still changing the DNA. It's just not quite as clean of a process. So yeah, they've been doing that for a long time, but it's higher tech now than it was.
We talked about cloning things.
Nice. Anything else you guys get? I know last group.
We talked about like GMOs, but I feel like Brian already said that and stuff like that.
Yeah, so GMOs is when they actually go in and specifically change the DNA to change an organism. So when we talk about biotechnology, there are a couple of different terms that are really similar. They all kind of overlap. I am never going to test you on the specific distinctions between these, but you have these definitions in your notes. DNA technology is specifically things for sequencing, which just literally means what is the order of the bases, the A's, T's, C's, and G's in that section of DNA, and manipulating and changing DNA. Biotechnology is trying to manipulate organisms or parts of organisms to make some kind of useful product. So when you're talking about like breeding apples or something, you're talking about biotechnology. And genetic engineering is direct manipulation of the genes for a practical application, so something like gene therapy. And like I said, these all overlap, like there are elements of genetic engineering in most biotechnology. There are small differences between these, so I just wanted you to see that, but we're going to kind of use all those words interchangeably as we talk about this stuff, this technology, in this class. These can be used for many, many different things. And you mentioned some of them. You mentioned things like GMOs. You mentioned things like therapeutic uses, like gene therapy. We can also use some types of biotechnology to manufacture proteins we need, like insulin. They can grow insulin proteins using gene therapies, and then take that insulin and inject it into people who need insulin. It can also be used for things like crime solving, forensics, paternity testing. We'll talk about some of that because since DNA is unique to the individual, you can use it sometimes as a way to identify or at least to rule out suspects if you have that type of evidence. And also for identification, you can look for certain mutations that cause diseases. They're actually doing a lot of investigation right now with COVID to sequence the genomes, like actually look at the genetic code of the different strains of virus that have appeared. That's a way they can kind of trace them back and figure out where new mutations might have occurred and kind of how the disease is spreading. They can actually look at the genetic code of the virus to track some of that as part of their research into how it's spreading, how it's changing, where it's going, where it might go in the future, all of that kind of stuff. So those are some. There are many other applications of this stuff as well. And as you know, this is a huge field in science right now. Macy mentioned like the medical field. This is a huge field of research right now in lots of different ways. And so there are more and more things that we could add to this list as we figure out more and more ways to use this. So this is the new slide I added today. If you want to kind of just jot this list down quick, it gives just a real brief description of the main types of technology we're going to talk about. We'll touch on a few others as we go through, but these are kind of the main ones that we're going to talk about in class. The first one is just DNA sequencing, which is literally just figuring out the order of the A's, T's, C's, and G's that make up the genetic code of an organism or just a specific section of a gene, if that's all that they're really interested in at that point. We're going to talk about the role of something called a restriction enzyme, which is used to cut up DNA by recognizing specific sequences. And some of these will kind of go together and you'll see why we would want to cut up DNA. Often this is used as part of a process to help identify where that DNA come from or who that DNA might belong to. We're going to talk about a technique called PCR, which stands for polymerase chain reaction. You don't need to write that down now because it's in your notes later. But that's a way to take a piece of DNA, say a sample from a specific organism or a piece of evidence from a crime scene, and make many, many, many copies of it so that you have a lot of copies of that section of DNA so you can analyze it. And this process called gel electrophoresis, which is the one that most of our labs center on because it's something that we can do in class, which is a way that we can separate molecules according to their charge, positive and negative charge, and their size. We, of course, are going to focus on separating strands of DNA because that's what our unit is mainly about. This technique can also be used to separate proteins of various sizes and other molecules, but we're going to focus on its use with DNA. Do you guys remember doing gel electrophoresis in 10th grade? You seen this before? This kind of thing? Okay. So we're going to review real briefly how it works. And then, like I said, we're going to be doing a couple of labs with this week. So we're going to watch, and this, I'm going to pull up a website. It has some sound effects that are kind of weird, but I don't know how to mute them without muting all of the volume for the people at home. So I don't want to do that. Hang on a sec. switch this to share this. Okay. So I just want to show you this. And I have to do this over here too. Well, the sound effects are not too loud today. That's good. Okay. And there are a couple of things that I want to kind of highlight in here. Mostly this is just going to be kind of a review of what this is and how this works, because we're going to be doing this over the next couple days. So it says we're holding a small plastic tube with some clear liquid in it. We've been told it contains DNA strands of several different lengths. And our job is to try and measure it and figure out how long those different segments of DNA are. And like I said, we'll be talking throughout the week about some different reasons that this is useful. It's going to make me do it on the computer. I think my smart board isn't working. There we go. So this is kind of what we want to do. And it says if DNA was shoelaces, we could sort them by hand into groups of different lengths and measure them. But we can't see it. There we go. Because it's super tiny and microscopic, so we can't see them. And that's why we use this thing called gel electrophoresis to sort DNA strands according to length. And we can do this with proteins as well. We're mainly going to focus on DNA. And so when we do this, we start with a gel made of agarose. This is kind of similar to the stuff that you grow bacteria on. And I have a piece of one. This one actually fell apart while I was making it. So it's like a corner of one. But I'm going to bring it around. You can touch it if you want to. It feels like Jell-O. But you can see it's got a bunch of little wells in it. And those little wells are just what they sound like. They're like little dips where you can put a little bit of liquid in there. And like I said, this is just kind of the corner of one. And I made several of these. You're going to be working with one in a little bit. By the time you see them, they're usually underwater or under the buffering solution. And so this agarose that makes up this gel works like a filter to separate these strands. And so you put the samples, and this is actually what you'll be practicing today, because as you saw, those of you that looked at it, the holes in that gel are really small. And so it takes a little bit of practice and a pretty steady hand to get the samples in there. And so that's what you're going to be practicing in a little bit. You put all the samples in one end, and then what we do is we run an electric current through here, because we sort this according to chemical charge and also according to size. Take just a second, think about what you know about the structure of DNA. Turn to the person next to you. Do you know what the electric charge on a molecule of DNA is? And see if you can figure out why.
Are hydrogen bonds positive or negative?
So here's where you start. What are the three main components of the nucleotides that make up DNA? Phosphates. What else? What are the other two parts? There's not water. Water helps put it together. Phosphate, sugar, that's the deoxyribose. What do you call the third part that sticks into the middle of the ladder? The A's, the T's, the C's, the G's. The nucleotide is the whole thing. That middle part is the base, the nitrogenous base. Yes? Sugar, phosphate, and a base. I know, we're back in school for real now, right? Okay. Phosphates, what kind of charge do phosphate groups have? Negative. Phosphate groups are negative. And so since the whole backbone of a DNA molecule is sugar, phosphate, DNA overall has a negative charge. And we use that fact in gel electrophoresis because the next thing we do is we put this in here with a negative charge on one end and a positive charge on the other, and this would go this way. But what that does is it causes the DNA to move, because if the DNA is here, it's going to be repelled by the negative and attracted to the positive. Okay? So the charge makes it move, but because the strands are different lengths, as they move through that gel, they have to kind of wiggle their way through. And if you have ever been in a very, very crowded place with little tiny people with you, do you know how quickly little kids can get lost in a crowd? That's what I always think of to remember how this works. The smaller strands can just kind of wiggle their way through this gel super fast. And so they travel the farthest. The longer strands are also moving, but they can't move as quickly through that gel. And so they don't go as far. So the sample of DNA you started with at the top separates itself according to length. And the shorter the strand is, the farther it goes. Yeah?
If you were to do it for a really long time, would the longer pieces eventually?
Yes, eventually they would get all the way through. So you do have to kind of stop. And usually these samples have like an indicator dye or something that you can see. So you can see like when the indicator gets to about here, you know your sample has kind of spread out nicely. Yep. So I think that's about all. Okay, so then as they move and separate, you get what's called a banding pattern. These are called bands of DNA that you can actually see, because what these little lines are is not just one DNA molecule, but it's a whole collection of molecules that are all about the same length. And so they've traveled about the same distance. So you can then kind of measure, if you have a sample where you know what the lengths are, you can compare the others and say, if we know this one is 100 base pairs and this one's 200 base pairs, we can estimate and say that's probably about 150 base pairs long. Does that kind of make sense? So that's basically how this process works. Yep, and then we're done with that thing. Okay. So what we're going to do today is we're going to practice loading the gels with the samples, because like I said, that takes a little bit of practice to find those little tiny spaces and be able to get just a little drop of liquid in there. So we're going to practice that today, and then we'll actually be running some like simulated experiments and a simulated crime scene later this week with some actual samples. We are going to kind of take some turns to do this so that we're a little more spread out. But before I show you and talk specifically about that, while you are waiting your turn to practice, and those of you who are at home, there are some pre-lab questions and some background questions that go with the lab we're doing tomorrow that you can also start working on today, okay? So in classroom, and I'm just going to switch over and kind of show you where this post is. This lab is posted under homework. And you'll see two attachments on here. One of them is the actual lab document that you have your own copy of where you're going to be typing in your answers. The other one is a background reading, and as you go through the lab, at one point it'll say using the background information in the lab and the additional reading, answer the question. So you will need to read through and reference this as well. But you should be able to answer questions up through, I think, like number 13 today as you start working on this, okay? So people who are at home, hang on just a minute. I'm going to come and show you what this lab setup kind of looks like and what they're doing here in a second. But you can open those documents and get started with that, and I will be back with you in just a second. Those of you who are here in person, we are going to, I need to line up my smartboard. I hate when it does this. Okay. So those of you who are here, pause for a second, look up from your computers. Thank you. There are 5 stations around the room, 4 on the lab tables, and 1 right over there. And what I'd like you to do at some point is come over to one of the stations, and I will hand these out in a second. You're each going to get your own one of these that you can use for class today. Oh, sorry. Let's throw that at you. is a micropipette. If you were here when we did the ELISA lab, we used these for that as well, kind of right before we left.
Yeah.
So what you will be doing here is you will be taking this, and in the beaker at the station, there are some micropipette tips. We're just going to use one tip for that station. Oh no. I have other tips, but these were the right size and they don't fit our pipettes. That's awesome. Okay, I will be putting out different tips that fit these. That's unfortunate. Once you have the tip on there, remember these are micropipettes. So this part moves up and down like infinitesimal, very small amounts. You will have a sample of food coloring, or it might be labeled practice loading dye. And all you're going to do is you're going to put the tip into the little vial of food coloring, take up just a tiny amount. When you do this for real tomorrow, it will tell you to do like 5 microliters, which if you look at the little plastic tip of the pipette, which you don't have yet, it's like half of the distance to the first mark on there. It's literally like 1 drop of liquid that you want to suck up in there. One thing I would recommend is that you, once you have the tip on your micropipette, you kind of reach in and like try and find, like wiggle it back and forth so it's like bumping against the sides of the well. Does that make sense? That's how you can tell the tip is actually in there. You do not want to stab it through all the way to the bottom though, okay? So this is a little, like, it's a little tricky thing. It's kind of fun when you can do it, but it takes a little bit of practice. So at the stations that you go to, you can try this like 5 or 6 times. There are way more wells in the gel than that, but several people are going to practice on the same gel. Does that make sense? The gels are submersed underwater because when you actually load them in the electrophoresis chamber, they are covered with a buffer solution, so you do have to do this when they're already underwater. If a little bit of the food coloring comes up and starts floating around the water, that's okay, but if a huge plume of color comes up, you had too much liquid in there. Does that make sense? Okay, so you can pull up those documents. Give me a second to find the correct pipette tips for this, and then we'll go around and we'll start practicing. Okay, and people who are at home, at this point, we're not actually setting up a lab today. Tomorrow, when we're actually setting up a lab, I will either put you in a group or I will show you the lab setup. But for today, they're just going to practice loading and continue working on that pre-lab. So if you guys want to stay on here and kind of work on the pre-lab together, you're welcome to do that. Otherwise, if you want to sign off for today, just go work on the pre-lab, read that background reading, work on some of those questions. We'll have a little more time to work on that tomorrow as well. But get a good start on it so you have an idea what we're doing when we get here tomorrow. Anybody online have any questions for me? You can either unmute or you can just type in the comments. All right, like I said, you're welcome to stick around while you're working, either to work with each other or to ask me questions if you want to. Otherwise, you are welcome to sign off for today. And we will see you in class tomorrow. Remember, Wednesday's class starts a little bit later because there's homeroom as part of the daily schedule. Any questions from anybody online? All right, we'll see you tomorrow if you step up. Otherwise, hang out if you want to.